Good afternoon, children. This is my second video explanation on the topic strategies for enhancement in food production. We started this topic last time and we studied definition of plant breeding, green revolution, the father of green revolution, and three very important tables I gave you to write and learn it by heart. This is very important from examination point of view. The disease resistant varieties of crops developed in India, then insect resistant plant varieties, another table, then list of examples of some Indian hybrid crops or the high yielding varieties, we call them HYVs. Then we introduced the main steps in breeding. First we studied here the objectives of plant breeding, and then we had the detailed steps. We discussed it under 6 headings, from germplasm collection for variability to evaluation and selection of parents. Then cross hybridization between selected plants, where important terms like emasculation and bagging were discussed. The fourth step was selection and testing of superior recombinants. The fifth was testing, release and commercialization of new cultivars. And the last was multiplication of improved seeds. All that we did in our first video on this topic. Today we will start with our second explanation on the very topic of strategies for enhancement in food production. Our topic today begins with mutation breeding. In your course, directly it says advantages of mutation breeding, but it's important to understand what is mutation breeding. Children, we know that if we expose the living cells to chemicals or radiations, they introduce some change in the genes, and that sudden change we call as mutation. So what is mutation breeding? It is the process of exposing. Here we are talking of plants, so we are talking of exposing the seeds to chemicals or radiations so as to generate mutants with desirable traits, right? And the same traits we can breed with other cultivars. In short, mutation breeding is a step to introduce the phenomenon of mutation in the plant genes, and the advantage is to get new varieties with desirable traits or characteristics. So let's move this with the definition. The creation of mutation at will and their utilization for production of new crop varieties is called mutation breeding. In India, it started since 1935. Let us see the advantages. The first advantage is great utility in crop improvement. Day by day, we are having better crops because of mutation breeding. Second, in mung bean, resistance to yellow mosaic virus and powdery mildew were induced. That means these two viral infections, right, they created a lot of damage to the mung bean, so that damage has been much reduced. The third advantage is a number of important varieties of crops have been evolved. Example, Sharbati Sonara, 64, Pusa Lerma, etc. The same terms I'll show you in your last class only. In the table that I gave you here, this is Sharbati Sonara. Alright, so they are the high yielding varieties that we discussed last time. Then, the next importance is resistance of yellow mosaic virus in okra or bhindi, that is transferred from a wild species and resulted in a new variety called Parbhani Kranti. So these are Indian varieties, therefore Hindi names. The semi-dwarf variety of rice, which is resistant to lodging, was obtained by a mutant gene. And also the mutation breeding helps in creating variability in self-pollinated crops. So a lot of variation which is normally not possible, but because of this breeding, it's very much achieved today. So these are the advantages of mutation breeding. Now we talk of some new terms, your syllabus only emphasizes on definitions. The first is heterosis or hybrid vigor. As the name tells, children, hybrid vigor, that means the hybrid of two pure line parents, right? Now, suppose this is parent A and this is parent B, and both are pure lines. One is having a desirable character here, but it's of pure species. Here, another parent with another desirable character. Now, the offspring that is produced as C, this has greater advantages when compared to either of the parents, right? Because the hybrid is better evolved over the parents in vigorosity. For example, it will be taller. C will be taller than A and B. It will be more sturdier or more strong and less resistant to and less susceptible to diseases, we can say. So therefore, it is better over the parents. Let's talk about the definition. The increased superiority of hybrids over the parents is called heterosis or hybrid vigor. The term was coined by G. H. Shull in 1914. Heterosis is practically very useful in improvement of all types of crops, and when two homozygous inbreeds of different genetic constitution are crossed together, the resulting hybrids are vigorous, productive, sturdier, and taller than either parents. That's what I said. C will be much better than A and B, and this is what we call as hybrid vigor or heterosis. Another term, inbreeding depression. Inbreeding means it's like a self-crossing, right? And somehow this is not very, very healthy. So that is also one reason to say that nature mostly favors cross-pollination in plants. But inbreeding depression, this, if we define it, it refers to the mating of more closely related individuals within the same breed for 4 to 6 generations. Thus, inbreeding depression is the reduction in the biological fitness of the individuals of a population as a result of inbreeding. So this is quite possible in animals and plants both, right? That inbreeding or the breeding between the more closely related individuals will not always be healthy, but in the long run, it continues to reduce the other qualities. Then, plant breeding for improvement in food quality. Now, in spite of so many achievements in the biological fields, let's say so much of experiments are being done day and night by various scientists, still it is surprising to know that around 840 million people all over the world, they do not get adequate food supply. Also, some 3 billion people, they suffer with micronutrient, protein, and vitamin deficiencies. Now, they have a term, we call them as hidden hunger. What does it mean? Deficiency of nutrients in a diet. Suppose a person is having a diet regularly from breakfast to lunch to dinner, but there is more or less some deficiency of some or other nutrients, then this will be called as a hidden hunger. That he's obviously having meals, but the supply of nutrients is incomplete. All right. So to talk on the same note, we have another topic called biofortification. What does this mean? This means breeding of crops with higher levels of vitamins and minerals or proteins or healthier fats, and this biofortification does not involve genetic engineering. Let us talk of some examples first. For example, let us talk of rice, right? Rice, we know, children, anyone who eats rice as the staple food, it is a very good source of starch. But imagine that today, because of so many experiments and innovations in biological fields, rice, if we eat, we have a choice to obtain iron fortified rice. That means eating rice is no more just a starch, but it will also give iron to our body, right? So this is what is fortify. Fortify means to make the food more complete by additional vitamins, minerals, proteins, and healthier fats. And definitely the products will be more healthy for the humans. Some hybrid crops with improved food quality that are developed recently, let's talk about them. Maize hybrids, they have been released in 2000. And maize, we know children, it's a source of protein, right? So this new hybrid has double amount of amino acids. Amino acids, we know children, they are the building blocks of proteins. So which amino acids? Double amount of lysine and tryptophan as compared to the normal maize hybrids. So obviously gaining or obtaining this hybrid for our nutritional value over the normal maize means getting double the proteins. All right. Then wheat variety. We have many sources of our food varieties that are dependent on wheat. Atlas 66. This has higher protein content and has been used as donor for improving cultivated wheat. So Atlas 66 means it's a wheat variety with more protein content. And the third is iron fortified rice, which contains 5 times more iron as compared in varieties that have been developed recently. So that means by name, if we talk of Atlas 66 or the maize hybrid or the rice hybrids, they have better fortified food values than the normal varieties. There's a body in New Delhi, IARI, Indian Agricultural Research Institute. It has released the following in the markets: vitamin A enriched carrots, spinach and pumpkin, vitamin C enriched tomato, mustard, bitter gourd, etc., and calcium and iron enriched beans, garden peas, etc. So this is what has been the result of various breeding experiments. What are the objectives of breeding for improvement? They aim to improve the protein content and quality. That means not just quantity but also the quality. Oil content and quality, they should be the healthy oils. Vitamin content and the micronutrient and mineral content. So till here we complete the topic on biofortification. It's an important topic and can be asked for some short notes or explanation. Maybe what is Atlas 66 or some other related questions. Coming to another new topic is SCP or single cell protein. Now, why single cell protein? First understand this. Normally, suppose this is a cow of some 250 kilogram weight. So the food of cow every day is some 20 to 30 kg of food. And a cow produces daily some 200 grams of protein. That's it. So out of 20 to 30 kilograms of food that were fed to a normal ordinary cow of 250 kilogram weight, the production net output is 200 grams of proteins only. The same if contradicted, just see the alternative of this. There is a microbe called Methylophilus methylotropus, right? This is a bacterium, and around 250 grams of this microbe, it can grow on waste materials. And can you imagine the output of this? Every day will be 25 tons of proteins. And why this so much of high content? The reason is that this has a very high rate of multiplication. So what is better, children? Industrially, if we want everyday supply, is 200 better in grams or 25 tons of proteins? This is much, much higher and beyond our imagination. So this is where we can say that protein supply, if suppose the entire human population is dependent on the cow only, then they will be not getting the regular supply. So this is what is single cell protein. That proteins can be also obtained from microbes, right, which can be generated on waste material. So there are dual advantage. Number one, there is cleaning of our ecosystem or our natural bodies, right, at the same time higher protein production. So that's what we are going to talk in single cell protein. Some more than 25% of human population is suffering from hunger and malnutrition, right? And today, if we talk of our vegetarian diet and compare it to the non-vegetarian diet, then the figures are something like this. Around 3 to 10 kilogram of grains are used to produce 1 kilogram of meat. Suppose we are non-vegetarians and we are going to purchase 1 kilogram of mutton or chicken. In that case, always remember, to make this 1 kilogram of meat, some 3 to 10 kgs of grains have been fed to the animal. So therefore, single cell protein is one of the alternative sources of proteins for animal and human nutrition. We said alternative. That means regularly we are eating the plant proteins and animal proteins in our diet, but this is as an alternate protein supply. The term SCP or single cell protein was coined by Carol L. Wilson, and the definition says dried microbial cell biomass or total protein extracted from pure microbial cell culture like algae, bacteria, filamentous fungi such as fusarium, graminearum and yeast. Now they grow on agricultural waste and they are used as food supplements for humans and animals. So children, I told you that they grow on waste substances and the outcome of protein is really very high. So what all things can be made to grow? Algae, bacteria, fungi, yeast, etc. Now some common microbes as SCP producers, you have to learn this by heart, children, right? And it is important that what all bacteria or which yeast, etc. The cyanobacteria, which can be used and especially used in nowadays, is spirulina and chlorella. Bacteria is Methylophilus, Methylotropis. The yeast is Candida utilis and filamentous fungi is Fusarium graminearum. So all of them, they have been used as microbes in SCP category for producing the alternative protein supply, right? Then let us talk of this spirulina. Spirulina, here what we saw as the cyanobacterium, is the microbe, right? And usually this is grown on, suppose this is the spirulina. Now this is utilized as a single cell protein. Why it is? Because this contains large amount of proteins, minerals, fats, carbohydrates and vitamins. Spirulina tablets are often taken as food supplement. And see how it is grown. It is grown on low cost substrate like wastewater from potato processing plants containing starch, straw, molasses, agricultural waste, sawdust, animal manure and sewage. So all this waste, when it is used by spirulina growth, then naturally environmental pollution is decreasing. So double advantage, as I told you, that the pollution is reducing and at the same time the spirulina, which is so rich in these contents, can be definitely used to benefit the human population. So in these 2 pages we discussed this, children. Let us go once again back to what I have done today. We started with mutation breeding, definition, advantages, definitions of heterosis and inbreeding depression, right? Here there's another alternative definition of mutation breeding. So one, either this here or this. You may learn any of them, whichever is easy to understand. Moving further, we started with a very important topic of biofortification, some hybrid crops, varieties in maize, wheat and rice, especially Atlas 66, then the iron fortified rice, then objectives of plant breeding and single cell protein, the definition, some sources. This is to be totally learned by heart, children. This is very important. Once again, I'll say there are direct questions, right? And then a comparative view between a cow and same weight, 250 gram. This was 250 kg of the cow and this is only 250 gram of the microbe and the amount of protein production. Okay, now let's move to the uses of single cell protein. Single cell protein, we saw how it is made and what are the microbes used. Let us see the advantages. The first use is single cell protein can be used as protein rich supplement of human diet. It will help to bridge gap between demand and supply of protein for human diet and reduce the pressure on agriculture. As we know, children, population is rising day by day. So obviously there's a pressure or a pressure on the food resources and agricultural practices. The same has to be dealt with very carefully. Use of agricultural waste and industrial effluents used in raising single cell protein will reduce the environmental pollution. So this we have completed here, right? Then we start with another new topic called as plant tissue culture. In the syllabus, children, it says plant tissue culture, the techniques and the applications. Today we'll only talk about the definition and techniques. All right, definition. See, usually before we come to definition, understand what is done. Plant tissue culture is done in this way, that a small plant tissue is used and it is called explant, right? It could be from the root tip, shoot tip or the shoot bud, leaf, petiole, anther, ovule, ovary, embryo, inflorescence, anywhere of the plant, right? And this is called as explant. It is brought into a laboratory under sterile conditions, that means germ-free conditions, and introduced into a nutrient medium. This nutrient medium could be a solid, semi-solid or a liquid. Now what happens? Single piece of this explant what we have brought will get the nutrition and will start growing into a bigger mass. In around 2 to 3 weeks, it grows into an unorganized parenchymatous mass of cells. We call it callus. And this specially is possible in 2 to 3 weeks in agar gel medium, right? Agar gel medium is a nutrient medium. This is usually supplemented with growth hormones like auxin 2,4-D and cytokinin BAP, that is benzyl aminopurine. This is one choice, that we took the plant tissue, brought it into the nutrient medium and it grew into a callus, but in a solid medium. Another option is that we introduce the explant into a, let's say, a liquid medium, and this liquid medium is supplemented with the growth hormone auxin 2,4-D. This is a better option, children. If we grow it on a solid medium or a liquid suspension, this is a better hand advantage. Reason, because it can be kept into a we can say a device where it can constantly be agitated or mixed at a 100 to 250 RPM. That means rotation per minute. So it can be put up into a machine and can be continuously made to shake, right? So as why? This will constantly mix the medium, break the cell aggregates into simpler groups, right? Smaller groups, and aeration of mixture is brought continuously. So suspension culture, as we call this, is growing faster from callus culture. So what is tissue culture, children? You saw it, that a technique of growing plant parts, tissues or organs into sterile culture medium under controlled environmental conditions, right? So this is of a much better variety of our choice possible of many uses that we'll discuss later, children. So understand, tissue culture is culturing of plant tissue into sterile medium under laboratory conditions, right? Either on a solid medium or a liquid medium. In your books, if I show you, this is at page number 517. This is shown tissue culture. You can see this. The explant in agar, the solid culture, and the explant that is being used into liquid culture we call as suspension. And as I told you, due to constantly being shaken, the cell aggregates are broken into small masses, and this is a better growth. So in a similar time, see the growth of callus, the unorganized mass, this is faster over the solid culture. Then finally, what is the next step, children, is subculturing. Subculturing means the culture has to be, now see what happens. The tissue culture cannot be maintained like this for a long time, right? So some of the cells, they should move to a new culture vessel like this. From the original culture tube, the pieces of callus, they are transferred into fresh medium after 4 to 6 weeks. That means around 1 or 1.5 months, they're transferred to new culture medium. So some culture pieces or cells, they are transferred from the main culture to the new culture vessels with fresh medium. And it involves various steps in the procedure. Let's talk of various steps, children. The first step is selection of explant. That means what you're going to culture should be definitely with desired qualities, should be very wisely selected, and it should be any of these: root tip, shoot tip, leaf, petiole, anther, ovule, ovary, etc. Second is sterilization, to make the plant part selected as totally germ free. Alright, for this we have two choices. First is surface sterilization and the other is complete sterilization. Surface sterilization is done for the living cells of the explant, and we treat it with chemicals like chlorine water or sodium or calcium hypochlorite solution. And the second set is for complete sterilization of the non-living equipments. For example, the instruments, their vessels, culture media and various instruments to be used. They sterilize with steam, dry heat or autoclaving at 120 degrees for some 15 to 20 minutes under high pressure. So this completely kills the germs of the non-living articles. The third step is transfer of explant to the culture medium. This is what I told you. This could be of any of the choice, into solid or semi-solid or the liquid culture. But remember, liquid culture has more advantages to be used. The fourth is callus formation, which is an unorganized mass of parenchymatous cells. And the fifth is organogenesis. That means formation of various organs and plantlets. I'll show you here. From your page number 19 textbook, you can see growth of callus into plantlets. In an agar medium, the explant is growing into tiny miniature plantlets, right? And this is how it can be transferred later to the fields. So this is what the organogenesis, we can say the leaves, the stem, the roots have started to form. Alright, the sixth and the last step is, the fifth and the last step was organogenesis and formation of plantlets, right? And if we talk about the uses of this culture, if you ask me why this entire process to be carried out in such a complicated way, it's not complicated, but yes, it is to be dealt up very seriously. The uses are, we can obtain cell biomass which is being used for biochemical isolation. We can regenerate these plantlets from the callus. That means what we saw here, we can regenerate the plantlets from the callus. Alright, then we can produce transgenic plants. Today a lot of transgenic plants have been obtained, right, which normally cannot be produced by sexual reproduction, and also isolation of protoplast, right? Then if we talk of regeneration of plantlets, this is our last topic for today. We have three choices. First is shoot and root regeneration, second somatic embryo regeneration, and the third is meristem culture. In shoot and root regeneration, the callus is treated, if it is treated with cytokinins like BAP, that is benzyl aminopurine, it will regenerate the shoot. Or if the callus is treated with auxins like NAA. What is NAA? I have written here. It is naphthalene acetic acid. In that case, the roots will be regenerated. The second is embryo regeneration. A somatic embryo. Somatic means from the vegetative cell of the plant body. A somatic embryo can be regenerated from a somatic cell by application of auxin 2,4-D. Finally, if we talk of meristem culture, this involves development of shoot systems and regeneration of adventitious roots from the developed shoots. So this is all about the introduction to tissue culture, definition and the technique. Now let us see the pages that I have completed today, because we have come to an end of this topic for today. We discussed mutation breeding, then benefits or advantages of mutation breeding, definition of hybrid vigor or heterosis, inbreeding depression, biofortification, the plants which have been recently developed with improved food quality, single cell protein, the sources of single cell protein, then uses of single cell protein, and tissue culture technique, the definition, then what is an explant, then technique or the steps that are involved, what is subculturing, what is the entire procedure in 5 points, especially with the emphasis on sterilization, difference between surface and complete sterilization, and we end up our class today with regeneration of plantlets under the 3 headings discussed. So that's all for today, children. I'll be sending you these 5 pages, some 5.5 pages. Take this as just one paragraph. So this is 1, 2, then 3, 4, 5 and 6. So I'm sending these pages. Kindly make up your notes and at the same time read the textbook, because next time, that is tomorrow, I will start with the applications of tissue culture. It will be definitely better, children, if you give a reading of applications so that you understand the topic in a better way tomorrow. Thank you.